Enzymes are catalysts that evolved in nature to make all of the molecules which are essential for life. It took nature billions of years to evolve the enzymes to catalyze many diverse reactions under very mild aqueous conditions. We would like to use enzymes for the more sustainable and clean synthesis of pharmaceuticals and other valuable compounds.
We find new enzymes by searching the genomes of plants and microorganisms using computational tools. We search for enzymes that are involved in bioactive natural products. These can have antimicrobial, anti-cancer and other biological activities.
Having identified the enzyme, we usually test them in reactions with a range of natural and synthetic substrate. Now, sometimes we found that this enzyme requires improvement in their properties. And to do this, we use automated method to engineer this smaller enzyme using directed evolution. And that way we can generate large numbers of mutated enzyme that we could use to screen for these improved properties. There are larger and more complex enzymes out there that can be difficult to engineer. So to address this, we developed gene editing method to engineer this larger enzyme in bacterial cell using CRISPR-Cas9. Now this allows us to produce complex molecule by fermentation, including antibiotics that can be difficult to be produced using full chemical synthesis.
We're most interested in enzymes that construct amide and peptide bonds because amide and peptide bonds are found in many pharmaceuticals.
We are particularly interested in enzymatic routes to new antibiotics to combat antimicrobial resistance. We find that there's very few antibiotics currently in drug development.
Enzyme can also provide a more cost-effective route to anti-infective agents that are urgently needed to treat neglected diseases in some part of the world.
Our work is highly interdisciplinary. It spans from chemistry through enzymology all the way to microbiology and genetics. We're particularly keen to train the next generation of scientists so that they can work across the disciplines.
We collaborate with pharmaceutical companies, biotech companies and other chemical companies who help to train our team and to focus our research towards commercial and societal impact.
Now, my research project here in Manchester involved engineering enzyme to produce antibiotics. Now, this is being carried out in collaboration with the company GSK. So after Manchester, I moved on to take up a position at GSK. So I guess the training that I received here in Manchester has really helped me in my career.